Thank you very much for the really kind introduction, and hello and good afternoon to everyone. I'm looking forward to spending the next hour with you all talking about some science. And I have to confess, it's probably going to be what I'll show you is less exciting than having a dog and a baby at home in the middle of a pandemic, but hopefully this will still be somewhat interesting. So the entire focus of my talk today is going to be around this notion of dosage sensitivity, which this is going to be familiar to many of you, I'm sure, especially those of you working in the clinical genetics space, but I wanted to get some primer material and just get us all on the same set of definitions. So what is dosage sensitivity? Well, for the purposes of this talk, dosage sensitivity means the intolerance in the human genome to changes in copy numbers. So for autosomes, this is, everything's preserved at copy number 2. So going down to copy number 1 by deletion could lead to haploinsufficiency if that loss of that locus results in a severe or deleterious phenotype. And conversely, gain of an additional copy of DNA at a given gene or locus can lead to triplosensitivity, where the duplication or gain of additional copies of a locus has a similarly deleterious phenotype or hit on organismal fitness. One area of focus for Mike Talkowski's group, which as Dr. Brownstein mentioned, that was where I just recently finished my PhD. But one core focus in the Talkowski lab is neurodevelopmental disorders like autism and intellectual disability. And in fact, this is actually a phenotype and a set of disorders for which we already know that dosage sensitivity is quite important. So specifically rare large copy number variants like deletions and duplications have been shown, now for the better part of 2 decades to be strongly associated with risk of neurodevelopmental disorders. However, we know that not every gene in the genome is equally sensitive to decreased copies or increased copies due to deletion or duplication. And so, of course, for any number of genes, certainly on the order of hundreds at this point, we know that loss of gene function due to deletion or point mutation confers a specific predictable autosomal dominant disease phenotype, like Coffin-Siris syndrome associated with loss of function of SMARCB1. And conversely, there are genes, a handful, maybe a few dozen, for which we know that gain or duplication of that gene results in a nearly dominant, quasi-dominant phenotype, like epileptic encephalopathy associated with KCNT1 duplications. But for genes like these, the reciprocal copy number change, so either duplication of SMARCB1 or loss of KCNT1, is observed in the general population, doesn't appear to have any strong effect on human phenotypes, to our knowledge at least. However, there's even this weirder subset of loci, like famous reciprocal genomic disorders such as the 16p11.2 microdeletion and microduplication syndrome locus, where we observe mirror phenotypes, where the loss of that locus leads to one subset of phenotypes. So in this case, macrocephaly, obesity, overweight, things like that. And the gain of that exact same segment of DNA leads to the opposite set of phenotypes or mirrored set of phenotypes. So gain at this locus, for example, results in microcephaly and low BMI. And so these are all really interesting patterns, but one question that we hope to answer in the study I'll be talking about today is, why are they the case? Why do we observe these different patterns of intolerance to deletions, duplications, neither or both? So a longstanding goal of ours to answer this question has been to map dosage sensitivity in the human genome. We're taking a multi-technology approach to this in the long term. So this involves projects like the Genome Aggregation Database, or GNOMED. This involves, exome sequencing efforts in the Center for Common Disease Genetics and various other consortia. And this involves also copy number variation aggregation from chromosomal microarrays. In the near future, we'll be putting all of the data I'm showing down at the bottom half of this slide together, but that's still work that's ongoing. So for today, what I'm going to be talking about is the one arm of this project that is wholly complete at this point. And in this study, which we recently published at the tail end of my PhD earlier this year, we aggregated rare large copy number variant data from chromosomal microarrays across nearly 1 million individuals, and used this to map dosage sensitivity throughout the human genome and across 54 different disease phenotypes. We'll talk about phenotypes in a second, but just starting with sort of the data set and the technical properties of the data itself, chromosomal microarrays might seem kind of trite or outdated by 2022 standards, but I do want to make sort of the devil's advocate case here that for the better part of the last decade or so, chromosomal microarrays were really the only human genetic data type that we had in sufficient sample sizes to do really robust, quantitative mapping of traits, especially in rare diseases. Now genomes and exomes are really becoming much more popular, but while we were working on this study over the last half a decade, we were able to access, chromosomal microarray derived copy number variant data from over 17 different sources, ranging from, population or nation scale biobanks like the UK Biobank and the Estonian Biobank through, array CGH run in clinical genetic diagnostic labs like GeneDx or Signature Genomics, all the way up through case control sort of disease mapping consortia like the Psychiatric Genomic Consortium or the Epi25 Collaborative. And so all of these, different institutes or centers or consortia have run copy number variant detection on chromosomal microarrays. And in total, we were able to aggregate nearly 1 million individuals worth of data with some technical limitations. Namely, we filtered these data quite heavily to only consider large, so greater than 100 kilobase, deletions or duplications appearing at less than 1% frequency in the general population as best as we could possibly assess it, looking at, genome sequencing catalogs, things like DGV, Decipher, and so on and so forth. And focused for all purposes of the analyses I'll be presenting today on the largely unique regions of the genome. So throwing out things like low copy repeats, segmental duplications, unmappable sort of satellites and the like. So this is going to be 95% of all protein coding genes in the genome are in these regions, but it kind of weeds out a lot of the technical issues that you might expect to see with copy number variants from microarrays. So now to get onto the phenotypes, like I mentioned, we aggregated data from nearly 1 million individuals across 17 different sources. And these sources did range quite a bit in the level of phenotypic granularity available to us. You could imagine full, electronic health record data from UK Biobank up through, in the case of the Epi25 Collaborative, that international consortium is focused on epilepsy genetics. And so they have detailed phenotype information about epilepsy, but they may not, be as well ascertained for other phenotypes that are kind of tangent to their primary research focus. So to account for this, what we did is we took all of the phenotype information we had available to us on a cohort by cohort basis, mapped that information onto the human phenotype ontology, which is essentially a structured way to represent different disease phenotypes in humans that goes down by, organ system and then tissue and so on and so forth. And in so doing, we were able to use sort of a hierarchical kind of clustering and pruning approach. I'm happy to talk about this more in the Q&A, to define a set of 54 disease phenotypes for which we were able to define, a superset of about 500,000 samples matching one or more of the disease phenotypes. We'll talk about the spectrum of phenotypes in just one second. And then in addition to these 500,000 cases, you can think of them, we were able to define 500,000 individuals for which, to the best of our knowledge, again, this is a limited, necessary step in the data processing, to the best of our knowledge, we had 500,000 individuals that were effectively controls and had no indication of any of these 54 disease phenotypes. And just one note, getting back to the neurodevelopmental disorders bent of this talk, we expect that the sort of under ascertainment of, undiagnosed neurodevelopmental disorders in large population data sets like the UK Biobank, for example, to be pretty low. These controls are largely sort of, adults, relatively healthy, or at least healthy enough to sign up for, a national biobank in Estonia or the UK Biobank or what have you. And so we don't expect false negative cases lingering in our sort of control pocket to be a particular problem for the phenotypes that, rare CNVs are going to be most prominent in. But to sort of prove this point, one of the first things that we did from this data set was look at every single one of these 54 different disease phenotypes. You can see on the x-axis here, the text might be a little small, so I apologize for that, and essentially ask the question, at loci where we know rare copy number variants contribute to human disease. So these are loci like the 16p11.2 microdeletion or microduplication I mentioned earlier, and similar genomic disorders associated with large CNVs. We can simply ask the question, in our sort of agnostic data-driven classification of different individuals into these phenotypic buckets of either case or control, do we see empirical evidence that these presumably pathogenic genomic disorder CNVs are enriched in the cases over the controls? And when doing this across all 54 different disease phenotypes, you can see deletions in red, duplications in blue, and combined deletions and duplications in purple. This color scheme will be consistent through the rest of the talk. You can see that the answer simply was yes, as expected, for about 80% of all of the 54 disease phenotypes, which do preferentially skew towards, again, as I mentioned, neurodevelopmental or congenital anomalies. We see an enrichment in the form of a odds ratio greater than 1 of cases carrying these genomic disorder CNVs compared to our sort of unaffected control group. So even though there are limitations, this is a pretty strong positive control, we felt. And then you'll notice that the effect size of these established pathogenic genomic disorder CNVs was not uniform across all the different phenotype groups, again, exactly like we expected. So to kind of get into the question of what phenotypes do we expect to harbor additional signal of association from rare CNVs outside of these known, pleiotropic syndromic genomic disorder loci, we excluded all of the CNVs that are accounted for in this top panel, and then just looked at deletions of loss of function constrained genes. So these are genes that have population level data to not that we believe to not tolerate loss of function in general population data sets like the Genome Aggregation Database and NOMAD that many of you I'm sure are familiar with. And then when we do the exact same analysis there and ask, what is the average effect size of a large rare CNV in our data set in cases versus controls for these loss of function intolerant genes or these constrained genes outside of known pathogenic CNV loci, we can see that there's a subset of, maybe a third of all of these phenotypes that have particularly strong effects where, the odds ratio arbitrarily chosen here of greater than, 2 to 1 in cases versus controls for rare deletions of these constrained genes. And so this is just a sort of simple lumping and splitting approach that we did to kind of distill down to the subset of phenotypes with that are most loaded for large rare copy number variants that we can focus down on in our subsequent analyses. And as you can see on the x-axis here, this is going to be, again, consistent with a lot of developmental disorders. The top phenotypes here being skeletal disorders, intellectual disability, growth disorders, weight disorders, facial abnormalities, and the like. All phenotypes that we fully expect to be loaded for, moderately to highly penetrant large rare copy number variants in their etiology. So, like I made the case, not only do we now have a subset of 500,000 cases and 500,000 controls across, many different phenotypes, but we can further stratify these cases into a subset of 17 out of 54 phenotypes with strong rare copy number variant effects. These are largely developmental phenotypes, but not all. And then we can subset out an additional 37 phenotypes for which there are sort of more modest, CNV effects. And these, by and large, if you go back to this last slide, you can see on the far right here, these are things like gout, arthritis, anxiety, atherosclerosis. They're phenotypes that maybe there are contributions from large rare copy number variants, but they don't pop out quite as prominently as sort of early onset pediatric developmental disorders. And then again, the one nice feature of the HPO or human phenotype ontology I alluded to earlier is it also allows you to stratify by affected tissue system. So specifically for neurodevelopmental disorders, which is an emphasis of our study, we can subset further into 6 HPO terms with strong rare copy number variant effects defined as above in neuro or neuroadjacent tissues or organ systems. And so this just essentially allows us to classify our cohort into different buckets for downstream analysis. With the data curation and study design out of the way, we then applied a series of different analytical approaches to quantify and understand dosage sensitivity throughout the human genome. This is essentially a little snippet of a graphical abstract, you can think of it, showing the different approaches that we tried. In the interest of time today, I'm just going to focus on a couple of these, but I do want to point out that you can go and look at our full paper online. It should be, I think it should still be open access, and if not, please email me and I'll send you the PDF directly if you want access to the full paper. But just for today, I'm going to focus on a couple of analyses that I think are some of the most interesting to come out of our study. And the first of these is genome-wide identification of new disease-associated CNVs. And so how we did this is we conducted fixed effect. We stepped across the entire autosomal human genome in 200 kilobase windows, stepping in 10 kilobase increments. So this is something like 270,000 individual windows we're testing for association between case and control for deletions and duplications. And we did this using a fixed effects meta-analysis across all of the different cohorts, the 17 different sources that went into sort of our overall study cohort. And this is very, analogous to what you might do in a conventional common variant GWAS. So you can visualize these data and think of these data just like you would a GWAS of any other trait. And so what I'm showing here is just one of our example phenotypes. So this is, again, neurodevelopmental disorders, the HPO term that most closely matches general neurodevelopmental disorder. And on the top in blue is duplication association. On the bottom in red is deletion association. And you can see that we get a number of peaks for both deletion and duplication in this phenotype that surpass a strict genome-wide significance threshold comparable to genome-wide significance in common variant GWAS. We did this for both deletions and duplications for each of the 54 different phenotypes in our study, each of the different 54 HPO terms that we just talked about. And when collapsing down all of these associations into distinct loci associated with where each locus is a specific association at a given position in the genome with either deletion or duplication and one or more diseases, so this is collapsing down across all 54 phenotypes, we identify 178 CNV segments associated with at least one disease at a false discovery rate greater than 1%. Half of these exceed genome-wide significance, but we also include another half of loci between false discovery of 1% and genome-wide significance, just sort of as a, suggestive or exploratory data set. Again, very consistent with some of the standards used in conventional common variant GWAS. So just two positive control data points to hopefully convince you that the sites that we're pulling out from this analysis are bona fide CNV disease associations and not just technical false positives. We first, using a genome-wide permutation approach, where we took these 178 CNV segments and randomly reshuffled them 100,000 times throughout the genome and asked, would we overlap known previously reported pathogenic genomic disorder associated CNVs more than we would expect by chance? And the answer there was a resounding yes. These 178 CNV segments that we're blindly discovering in our data set of about 1 million individuals with genome-wide meta-analyses were 7-fold enriched for previously reported genomic disorder CNVs curated from a variety of different sort of more authoritative literature sources like either ClinGen or previous publications or Decipher or what have you. And so this really suggests to us that we're recapturing a lot of known signal. But even beyond the level of known pathogenic CNVs, even if you match into OMIM on, we have HPO terms, you can use the HPO terms to match directly into OMIM for genes that are known to be associated with that exact same HPO term. When we do that in a similar permutation approach, we find a modest but highly statistically significant enrichment for individual disease genes within these larger CNVs, again suggesting that we're pulling out signal that might be informative and might extend beyond these large pathogenic known established genomic disorder regions. So with these 178 disease-associated CNV segments in hand, we next wanted to ask the question, what defines these segments? What defines these loci? What makes them different from, your random 500 kilobase chunk of the genome on chromosome 2 or whatever? And so using similar permutation-based approaches, we effectively resampled an expected distribution for the deletions and the duplications in our data set for a variety of different genomic features. This is number of genes, this is, expression of those genes, mutational constraint of those genes, and on, disease association of those genes. And when we did this, a few features really jumped out as being key distinguishers of the empirically disease-associated CNV segments compared to just a null background from the genome. And the first is that these empirically disease-associated CNVs were particularly gene-rich. So the average disease-associated CNV in our data set of 178 segments overlapped about twice as many genes as you would expect by chance when matching on the segment size. So if you have a 500 kilobase deletion, and say on average you expect, the average 500 kilobase chunk of the human genome to overlap, I don't know, 5 genes, 10 genes, something like that. Well, the deletions in the 500 kilobase deletions that were disease associated in our analysis were overlapping 10 or 20 genes, for example. So after conditioning on their increased gene density by matching on the number of genes, not just the size of the CNV, but also the number of genes in those CNVs, we still found that these CNVs landed on genes that were mutationally constrained beyond what you would expect just by randomly sampling genes from the genome. And so how we're measuring it here, just in one example, but this holds up for both missense constraint, loss of function constraint, other forms of mutational constraint or intolerance, is LUF, which is a metric derived from the genome aggregation database that I'm sure many of you are familiar with, but effectively quantifies how depleted each gene is in the general population for segregating rare loss of function point mutations. And so what you can see here, especially for deletions, is this bar represents, the observed value in our empirical data set for deletions associated with one or more diseases. And this background distribution shows that the deletions in our data set overlap genes that have lower LUF values corresponding with greater mutational constraint against loss of function than you'd expect just by randomly sampling from the genome. So two stories emerge here. One is that it's the number of genes in a CNV that correlates with its pathogenicity, but not only is it the number of genes, but it's also the nature of those genes. Kind of not surprising, right? It's, more genes, worse overall phenotypically, but also if you hit one particularly strong effect gene or, fully penetrant gene in a severe phenotype, you don't need to hit 20 other genes to manifest a phenotype detectable, in a case control analysis like we're doing here. Now, two more tidbits from this analysis just before we move on that I find kind of interesting is that it gets even more nuanced if you consider penetrance. So as a rough proxy, so our genome-wide fixed effects meta-analysis gives us an odds ratio estimate for each phenotype in each CNV throughout the genome. We can use this to stratify CNVs based on their rough penetrance using odds ratio as sort of a proxy. So if you take the 178 disease-associated CNVs we identified, and you split them into 3 bins, just thirds, ranking them by their highest odds ratio for any one phenotype, as a proxy for penetrance, segments that are highly penetrant and show up in the top third of effect sizes from our meta-analysis overlap genes that are under greater constraint on average than segments in the bottom third. So these are going to be more incompletely penetrant or sort of variably expressive CNVs that might be more risk factors. They don't tend to land on genes that are quite as constrained as these sort of highly penetrant, no doubt, autosomal dominant. You can think of them as almost fully penetrant autosomal dominant CNVs. The average odds ratio here, I think, is like 444. So yeah, you might have one control for every 444 or 443, cases that carry the CNV, but that's effectively fully penetrant. And that correlates quite strongly with evolutionary constraint on the level of individual genes. And then just one more result, again, sort of showing that the nature of the genes within these CNVs is important to consider, is that the number of genes in each CNV correlated positively with the number of phenotypes associated at the same statistical threshold in our meta-analysis. So larger CNVs hit more genes, and those genes tend to have their effects across more phenotypes, whereas, say maybe a focal CNV of just one or a handful of genes will typically, in our data set at least, exhibit association with just one or a handful of different phenotypes. So we reasoned that if the genes within these CNVs were the predominant feature that sort of dictated their disease association, we should be able to confirm this by looking at an independent data set of a different type of genomic variation, namely de novo point mutations as ascertained by whole exome sequencing. So for this analysis, we focused on neurodevelopmental disorders because to date, exome sequencing in neurodevelopmental disorders has been particularly prolific and productive in discovering disease genes, either in autism, for which we've played a role in the past as part of the Autism Sequencing Consortium, or developmental disorders more broadly, perhaps most prominently spearheaded by the Deciphering Developmental Disorders Project over in the UK. And so collectively, between the Autism Sequencing Consortium and the Deciphering Developmental Disorders Project, we were able to aggregate de novo mutation data for 46,000 individuals with neurodevelopmental disorders whose parents were unaffected. This comes out to just north of about one coding de novo mutation in each child's exome. And so this gives us a really powerful data set to intersect with our disease-associated copy number variants and ask, what are the patterns and properties of coding de novo mutations in neurodevelopmental disorder patients in these CNV regions? And so using a similar permutation-based approach that also matches not only on just the size of the CNV, the number of genes in each CNV, but the individual predicted de novo mutation rate in sort of under neutral selection, so no selection, in the general population, again, relying heavily on resources like Nomad for these permutation approaches, we were able to ask, in this combined data set of about 46,000 neurodevelopmental disorder trios, are there more de novo coding de novo mutations occurring in the genes within these CNVs in independent patients, independent cohorts, than you'd expect by chance under neutral mutation? And the answer was yes, with an asterisk. So you'll notice there's a trend here. The signal's kind of weak for deletions, largely because a lot of these, individual monogenic causes of neurodevelopmental disorders due to protein truncating variants, or PTVs, collapse into just a single gene. And so our large CNVs don't really capture the signal quite as well here. But it's nominally enriched. De novo mutations were observed at a greater excess than you'd expect by chance in our deletion-associated regions, but not our duplication-associated regions. And similarly, but conversely, when we look at missense de novo mutations, we found specific enrichments in duplication-associated regions, but not deletion-associated regions. So like I was alluding to, there's quite a long list at this point of genes that have been identified by exome sequencing and robustly implicated in neurodevelopmental disorders. So we then ask the question, are these signals just wholly explained by existing lists of known neurodevelopmental disorder disease genes as defined specifically in these cohorts? So not using external information from Decipher or OMIM or what have you, just genes that met the statistical threshold for association either with autism or developmental disorders in the two studies on the left. And then when you look at protein truncating de novo mutations in deletions, the signal goes away entirely. So this tells us that, yes, there is an excess of de novo mutations, de novo protein truncating mutations in genes in deletions associated with neurodevelopmental disorders, but those genes tend to be genes we already know about. And so there's probably not a ton more to find in the convergence of PTV de novo mutations and deletion copy number variants. However, what got really interesting is when we did the exact same analysis for missense de novo mutations in duplication copy number variants, we saw the signal, if anything, get stronger. And so this to us tells us that there's actually probably quite a bit of signal still to be captured in the intersection of missense mutations and duplications that may capture new strong effect neurodevelopmental disease genes not already captured by a lot of existing analyses that kind of focus more on haploinsufficiency and loss of function. So these are probably going to be genes that are implicated in neurodevelopmental disorders through either gain of function or altered function or some kind of different mechanism than haploinsufficiency, which I think is a pretty exciting future direction. Just to illustrate this a little bit, sort of if you think of it as a transpose, we can then look, at each segment and say, what's the total excess of de novo mutations in the neurodevelopmental disorder exome sequencing data set for each of these segments. And then we can essentially tally for each gene, what fraction of the total excess of de novo mutations is concentrated in each gene in this segment, and then rank those genes into sort of the gene with the greatest excess, the gene with the second greatest excess, and so on and so forth for all the genes in the segment. And if no one gene were important, you would expect these colors to be roughly uniformly distributed, roughly. There's going to be some sampling biases, and it won't follow perfectly uniform distribution, but you would expect more dark colors in this plot. And looking at it another way, if you sort of normalize all of these to a proportion of 100, we can see that for 38% of all of the deletions associated with neurodevelopmental disorders in our data set, the entirety of their excess of de novo protein truncating variants fell in just one or two genes. These are going to be sort of quasi-dominant, either fully penetrant or intermediately penetrant driver genes, individual genes within these CNVs. And then when you ask, well, let's just look at the top, dozen segments. What are these genes? I'm sure many of you in the audience are going to recognize these genes immediately as known autosomal dominant neurodevelopmental disease genes. However, for some loci, say the 1p36 terminal microdeletion syndrome, SKI emerges as the predominant gene here, but you can see there's a handful of other genes, same thing with MAGEL2 and HRNP and HNRPNU, at these other terminal loci, 15q11.2 is interstitial, but there's a situations that are somewhat rare where there's clearly, multiple genes that have excesses of de novo mutations, but in general for deletions, the vast majority of cases are a single gene harbors most of their de novo mutation excess for at least protein truncating variants. When we do the same analysis for missense de novo mutations, the signal is a little bit more muddied. And so here you can see, kind of like I was indicating, that if you didn't have a preponderance of single genes responsible for driving the neurodevelopmental phenotypes in these regions, you would expect to see more sort of the greens and blues and purples representing contributions from genes beyond the most burdened gene in this region. And this is exactly what we see. And so in fact, when you look at even the most burdened regions specifically duplication associated regions that have the greatest excess of de novo missense variants in neurodevelopmental disorder patients, you can see that sure, there are a handful of genes, like SMARCA2 is one of my favorites in particular, where that is a known association with Nicolaides-Baraitser syndrome that's been presumably caused by gain of function missense mutations. Well, we now also see duplication association at that locus in neurodevelopmental disorders. So again, that's kind of consistent with gain of function association at that locus. But for the other genes also incorporated in those duplications, we see excesses of de novo missense variants in those genes beyond what you would expect by chance as well. And so maybe SMARCA2 alone is not the only gene involved in this syndromic duplication. And I think this is really true, at least based on the data that we've looked at so far, for a lot of duplications. It's not going to land on just one gene. It might be, oligogenic contributions from multiple genes, which I think is particularly interesting, but kind of analytically challenging. So I'll pivot here for sort of the second set of results and say that one aim of our study was what we just talked about, which is identifying regions of the genome that meet some statistically significant threshold for association between cases and controls for, particularly severe or rare disease phenotypes like developmental disorders. This is good, but it really is limited by the fact that we have imperfect phenotyping, strict statistical thresholds, and limited case data, especially for rare phenotypes, and especially for rare phenotypes in organ systems that don't acquire CNVs quite as often as neurodevelopmental disorders. So we wanted to extend this data set as much as possible and come up with a way to predict the pathogenic effects of CNVs for every gene in the genome, not just those that meet exome-wide significance or genome-wide significance. And we reasoned that we had probably enough data to turn this into a machine learning or a data science problem, and less so a statistical genetics or genetic epidemiology problem. And so to approach this, we first started by defining training sets of about a couple hundred genes each, where we used existing evidence from the literature to define sets of genes that were either, we had pretty good confidence were haploinsufficient or deletion sensitive, or triplosensitive or duplication sensitive, shown here in red and blue, or conversely, you can think of sort of the inverse of this, of sets of genes that tolerated deletions and duplications. So, technically haplosufficient or triploinsensitive, but the terminology gets kind of confusing. So I'll be calling those mutationally tolerant genes. So we have these 4 sets of genes based on literature evidence, having not looked at our own data at all yet. We then computed for every one of these genes the effect size we observed in our meta-analysis, our gene-based meta-analysis, for neurodevelopmental disorder cases versus all the controls. And then we tasked a variety of 8 different machine learning models. Can you predict these effect sizes that differ between haploinsufficient genes and mutationally tolerant genes, or same deal for duplication-sensitive genes or triplet-sensitive genes? Can you predict the identity of these genes just from gene-level features themselves? This includes things like chromatin context, gene size, exon intron spacing, constraint, conservation, expression, protein structure, you name it. We tried to throw these features into the model. And the surprising result we were met with was actually most of these models worked pretty well at predicting our training sets. So these are receiver operating characteristic curves and precision recall curves, for those of you not familiar with looking at machine learning performance data. But effectively, each different colored curve here is one of the 8 models I mentioned. And the closer that model is to either the top left or top right corner, the better it's doing at predicting the identity of our training genes, either the literature-based haploinsufficient or triplosensitive genes, or similarly, the literature-based mutationally tolerant genes that probably don't matter in the case of copy number variants in disease. We then used this yellow model, which was sort of the principled averaging, you can think of it, as all 8 different models. It's known as an ensemble model. We used this ensemble model to define 2 new metrics that for every protein coding gene in the human autosome, across all human autosomes, the probability of haploinsufficiency, or p-haplo, and the probability of triplosensitivity, or p-triplo. When you line up p-haplo and p-triplo, so these are our haploinsufficiency or our deletion intolerance score, you can think of it, and our duplication intolerance score on the y-axis, again in red and blue, staying consistent with previous coloration, we observe right away that these 2 scores are correlated. And so on average, this is telling us that genes tend to either be sensitive to both deletion and duplication, or sensitive to neither. And yes, there are plenty of exceptions, hundreds of genes for which there's the exception. But in general, our model makes a prediction that genes will either be bidirectionally dosage sensitive, meaning sensitive to deletions and duplications, or sensitive to neither. And then again, of course, just as before, we wanted to do a variety of quality control analyses to make sure that we believe these scores, that they're not just garbage. And so we looked at several different analyses. I'm just going to show 2 on this slide. The first is we took sets of genes from the literature or from a variety of other sources that we had some prior to expect them to either be enriched for high scoring genes or low scoring genes in our model. And we see this for, there's a variety of gene lists shown here, where darker colors indicate greater enrichments, lighter colors indicate depletion. And this kind of lines up with what you'd expect based on the biology of these gene sets. And then again, we can look at external data sets. In this case, the genome aggregation database, which has, our previous work in that data set has defined structural variants from about 11,000 unrelated individuals from whole genome sequencing. And these cohorts are wholly independent of the array data sets that we're working with here. And so when you look at the rate of loss of function deletions or whole copy gain duplications per gene for all genes, in our model, in this NOMAD database, which is essentially a population reference, you can see that our model's higher scores correlate negatively with the rate of loss of coding CNVs in these genes in the general population. Again, consistent with the fact that if these genes really are sensitive to deletion and duplication, you should not see loss of function deletions or copy gain duplications of those genes in a general population database like NOMAD. getting back to the neurodevelopmental disorders angle, one of the analyses that we were most interested to pursue was, could we use these scores, the p-haplo and p-triplo scores, to help improve the interpretation of coding CNVs or short variants in exome sequencing data? And so what we did, this data has now been published. Jack Fu, a postdoc in the group, had a really nice Nature Genetics paper just recently from the Autism Sequencing Consortium on this data, but we've called rare copy number variants, de novo rare copy number variants in about 14,000 children with autism spectrum disorder and their 5,000 unaffected neurotypical siblings. And this is done from exome sequencing data. So again, wholly independent from the array data we're looking at here. And we asked the question, if we, even though the de novo deletion and duplication data in these data sets are sparse, can we help stratify the risk contributed by the top set of deletions or duplications from the bottom set? And empirically, we can show that, yes, these p-haplo and p-triplo scores tend to stratify with higher scoring genes carrying more risk for autism in this independent data set as compared by their the affected children directly to their unaffected siblings, both for deletions and duplications using p-haplo and p-triplo. Similarly, we actually find sort of a corollary to this, that these CNV-based deletion and duplication intolerance scores, p-haplo and p-triplo, can help to pinpoint genes that have greater excesses of de novo protein truncating variants or missense variants, shown in dark red and orange. And notably, these models are not just picking up background mutation rate, because you can see that the enrichment for de novo synonymous mutations is pretty flat, shown in gray. And so we've looked at this, and there's relatively limited added information in these CNV-based models beyond existing constraint models, like those available from Nomad, like LUF or PLI or missense constraint. But if you take the intersection of our CNV intolerance scores and the Nomad constraint metrics that are already widely used in the neurodevelopmental genetics community, that pinpoints the subset of genes for which we observe the greatest enrichment. Again, suggesting that if you want a really high confidence set of mutationally intolerant and dosage sensitive genes, using CNV data and short variant data like SNVs and indels is going to be a really powerful approach for further enriching your data set. One of the last two analyses I want to present is, these scores are all well and good, but we wanted to provide recommendations for thresholding these scores to define subsets of genes, so gene lists that we are willing to put our flag in the ground and say, this is our predicted set of however many duplication sensitive genes, this is our predicted set of however many deletion sensitive genes at some interpretable threshold. And so how we did this is, starting from our highest scoring genes and walking backwards to our lowest scoring genes in percentile bins on p-haplo and p-triplo, we asked for each of those bins of genes, these are about 100 some odd genes per bin on average, what is the observed effect size in our empirical data set of microarray data in neurodevelopmental disorder cases versus controls for those genes, and what is the cumulative effects. So the points are the individual bins of genes, and the solid line is the cumulative effect of taking all genes above some value on the x-axis, which corresponds to our p-haplo score here. And so we then wanted to titrate a cutoff that was approximately comparable to the effect size we observe for deletions of loss of function intolerant genes in our own microarray data, using loss of function intolerant genes defined in the genome aggregation database based on SNVs and indels, so not looking at CNV data to define those genes. And so using this sort of rough calibration approach, we define a threshold of p-haplo greater than 0.86, corresponding to about 3,000 haploinsufficient genes, where deletion of those genes has the same average effect size as deletion of a gold standard loss of function constrained gene. We can do the same thing for duplications. And the first thing you'll notice right away is that on average, the effects of duplications are weaker than the effects of deletions. This is fully expected. This is consistent with, both prior literature as well as our, previous analyses in this same data set. But the most interesting thing here is that if you zoom in on the right-hand tail of p-triplo scores, we're able to define a threshold of 0.94 or greater, being a cutoff that gives you about 1,600 genes where whole gene duplications have the same average effect size as loss of function of gold standard mutationally constrained genes. And so I think these 1,600 genes are particularly interesting as a starting point for, further biological follow-up and characterization, because these are genes that our model predicts would not tolerate whole gene duplications. And so the next question becomes why? And I think that's going to be a really interesting area for future discovery. So the last result I'll present here is sort of the final analysis we did in this paper. I think this is going to be figure 7 or 8 in the Cell paper if you want to go back to that. But we wanted to ask the question, getting back to genome biology, what makes these genes dosage sensitive? And so we asked this question 2 ways. The first way we asked it was what features at the level of individual genes were most predictive of bidirectional dosage sensitivity, meaning intolerance to both deletions and duplications. So we did this essentially by taking the minimum for each gene of its p-haplo and p-triplo score, and then just, a feature importance analysis, correlating it back against the raw gene level features we used in model training. This correlates pretty closely with the weights learned by the model. It's, we have a supplemental figure if you're curious about that, but this we felt like was a clear way to present the data. And the most prominent features predictive of bidirectional dosage sensitivity, or again, sensitivity to deletions and duplications, were existing constraint metrics. So this is NOMAD LUF, NOMAD PLI, the NOMAD loss of function missense Z-scores. These are other scores like, the loss of function selection coefficient from Casa et al. Nature Genetics, exon conservation in other species, the haploinsufficiency index from Matt Hurl's group back from 2011, ARVIS. These are all scores that already are in, wide circulation in the human genetics community, and our model is showing that they are broadly predictive of deletion and duplication constrained genes. And so here's just a couple examples of how these features actually look. You can see that both for NOMAD LUF scores, as well as, exon conservation as measured with FastCons across, mammalian species, this purple group is the highest. And these are genes that score greater than, 0.9 or greater than, say, 0.5 for a lower quality, lower confidence set, but this dark purple greater than 0.9, scoring greater than 0.9 for both p-haplo and p-triplo, are the most prominently enriched for these types of constraint and conservation features at the level of coding exons. We then asked the question sort of on the other axis, which is, were there any features that preferentially correlated with deletion sensitivity or haploinsufficiency, or triplo sensitivity or duplication sensitivity? And so we used a similar approach, except this time we're computing the difference between the p-triplo scores and the p-haplo scores for every gene, where a gene that scores very positively is going to be preferentially more duplication sensitive, and a gene that scores very negatively is going to be preferentially more haploinsufficient. We then did a feature importance analysis of these types of essentially residuals off the diagonal. You can think of it that way. And the features that popped out here were a little bit harder to interpret, but I think they do start to provide some inroads into understanding the differences between haploinsufficiency and triplo sensitivity. And so just sort of summarizing these features, again, this is all speculative and very preliminary results, but I think for the developmental biologists in the room, some of the features that correlate most strongly with haploinsufficiency but not triplo sensitivity are hallmarks of developmentally critical, temporally and spatially regulated genes. So these haploinsufficient specific genes are genes that are preferentially isolated from other genes, further from other genes. They tend to be a little larger. They tend to be surrounded by this complex network of bivalent or poised enhancers. These are all hallmarks of genes that need to be turned on in expression at a specific time or place, and then turned off in other tissues or other time points. Now, conversely, the features that we found associating most strongly with duplication sensitivity, but not but not deletion sensitivity, were genes that tended to be shorter, GC rich, and localized to gene dense regions, characterized by highly activating chromatin contexts. And so these are genes that we kind of struggle to put a pin into exactly what how to characterize these, but they do kind of match the description of a lot of these sort of short GC-rich transcription factors, like the forkhead box proteins or the GATA proteins and genes like that, that have broad transcriptional consequences when dysregulated. And for whatever reason, our model is saying that those types of genes tend to be the ones that are most sensitive to increased genomic copy number, but not decreased genomic copy number. And so that was a lot to digest, I'm sure. I'll wrap up there. I think we've saved hopefully some time for questions, but before we get to questions, I just really want to thank everyone who contributed to this study. Like I mentioned, it was a huge collaboration across 17 different centers, just a few of which I've listed here. There's many folks that contributed to this beyond the scope of this one slide. And so thank you for your attention. I'd be happy to take any questions.
That was excellent. Thank you. Thank you, Ryan.
No problem.
Yeah.
Please raise your hand so we know to unmute you, and let's start this off. In the meantime, I guess I'll ask a question. First of all, that was an incredible talk. I am so impressed, and I have a billion things that I want to ask you offline. But the one that really made me perk up my ears like an animal smelling something was your missense and duplication in the neurodevelopmental disorders that are left to be discovered. Can you give, because of the audience here, can you give any advice for researchers as they leave here and go back to their own cohorts and to investigate or follow up on this type of thing, because that sounds really exciting?
Yeah, it's an area that we're also really excited about. I think our assessment is that the data necessary to really make high confidence predictions of gain of function specific disease genes are still kind of coming online, and there's a lot of ongoing efforts to characterize those. But as a rough first pass, what we would look for is genes that score highly in our p-triplo score, for example, but not in our p-haplo score, and genes for which there's a strong missense constraint in population data sets like NOMAD or others, but not strong loss of function constraint. And that would be a really good starting point. That's going to be an imperfect classifier, but those are sort of the two signals that we would be thinking about. And then, of course, there's various models to predict the loss of function or gain of function effects of individual missense mutations. So if you had a mutation in a specific patient, it might be worth exploring those types of models. But I'm less familiar with sort of the performance of those. And so in broad strokes, that would be what I would start with personally.
Then to take it a step further, were there any CNVs that you were surprised or contradicted the commonly accepted pathogenicity of the CNV, where you know, you were kind of like, ugh?
Yeah, so there's actually, I'm going to take the time to go all the way back here, because I think this is a really interesting point that you raise. The answer is yes, there were a few that surprised us. I guess I can give one quick anecdote, is that for, our study here corroborates this finding, but we actually already observed this in an earlier study, the NOMAD, structural variant study that I alluded to earlier, which was just the population mapping from whole genome sequencing of all structural variants. Duplications at 2p13, which involve NPHP1, are about eightfold, if I'm remembering correctly, eightfold higher frequency in East Asian ancestry individuals compared to all other continental ancestries. And if you look at the literature for a lot of the, NPHP1 duplications have been, proposed as a potential genetic risk factor for autism and other neurodevelopmental disorders. And so if you look at those case studies that have proposed those associations, they're largely in East Asian patients and families. And so that analysis is kind of sort of a sobering one, that there may be ancestry specific risk factors here, or there may be risk factors that have been previously proposed that may be driven by population stratification in the individual case reports. And I'm not saying whether it's one way or the other, but it definitely does sort of raise caution for loci like that until we can really get to the bottom of what's going on there. So that's from NOMAD. But here in this study, why I went all the way back to this slide is what I'm showing in this scatter plot here that I kind of just blew through really quickly is, if the point is a square or a diamond, it came from our discovery analysis, doing this genome-wide association scan that I'm showing on the left. If the point is a circle, we didn't pick it up at our sort of robust statistical genome-wide thresholds in our discovery analysis, but it was present in one or more of these large reference data sets like ClinGen or Decipher or a few other, large-scale studies and surveys that have been published to date, looking at genomic disorder loci specifically. And so most of these literature loci, I think there was 96 in total, most of these genomic disorders from the literature, these circles, were present in our discovery set, but of those that weren't, and of those that didn't pass any statistical threshold in our data set, they kind of fall into two camps. The one is these points down here have lower odds ratios and don't have as strong of evidence statistically in our data set. And so these are the types of genomic disorders that, most optimistically are incompletely penetrant risk factors. And there's a list of, just for those folks who are curious in these particular points here, there's a list in the supplement of our paper of all of the points on this plot with the odds ratio, so you can go and pick apart which ones are these little, circles with the X's that we didn't include in our analyses in this paper. So these types of points are going to be either false positives, that we with larger data can now show they aren't really that strongly associated, or they're incompletely penetrant risk factors that we just don't have the sample size or the right phenotype, or specifically endophenotyping is potentially one confounder here. Like if it's these CNVs are associated with a very specific type of, let's say, renal malformation. But if you look at all renal malformations more broadly, they're not that strongly associated. That might show up as a weaker effect size here. So that's sort of the one camp where maybe our study can draw some skepticism into whether or not these are bona fide disease-associated CNVs. The other side is like the strongest single effect size we observed in our entire study. This has an odds ratio, this point here has an odds ratio of about 2 to the 15th. It's an absolutely astronomical odds ratio. But it doesn't meet genome-wide significance in our study because it's just so vanishingly rare in the general population that I think this was something like 6 cases carried this deletion and 0 controls out of 500,000. And so there, if I had to bet, this is definitely a real disease-associated CNV, and it's a very potent, highly penetrant locus, but it's just going to be so rare that these types of statistical association studies won't be able to pick those up. And so those are kind of the two types of ways in which I think our study might help clarify some pre-existing reports. But I do want to again emphasize that I think like 70 or 80% of all previously reported genomic disorders in ClinGen and Decipher and the like did show up in our discovery set. So most of what's out there looks to be pretty good to us.
Great, thanks. Actually, something you just mentioned, so it's interesting that you have almost 1 million samples here, right? And there's still things that are too rare to capture with those. Yeah.
That's the thing that I think genome and exome sequencing will help a lot, because these loci may be situations where very large CNVs that are able to be captured by microarrays are infrequent, but if you're able to zoom in at the level of like single exon deletions or single gene deletions, maybe we'll start to get more power to start to detect individual associations within these large regions. And so that's an area I'm particularly excited about thinking about, next steps.
Yeah, that was something I was going to ask is, you mentioned, combining CNV derived scores with like SNV and small ones are better. And is that just because now you're adding another 1 million samples, or because you're actually capturing different mechanisms? And it sounds like probably the latter.
Yeah, I mean, it's a really interesting question. It's probably both, to be honest. I think, sample size is the number one factor for anything basically statistical genetics. That's kind of always the rule, I feel like. But when we directly compare head-to-head SNV and indel constraint scores with, well, here, let me just, I know we're kind of over time, but I do have a supplemental slide that directly, looks at this that might be interesting. Here we go. So if you look at the correlation of our P haplo scores in red and P triplo scores in blue versus a variety of these sort of existing scores that are commonly used in the community, most of these scores are pretty well correlated. But for instance, if you look at let's say LUF, which is sort of, in my mind, one of the gold standards for defining loss of function constraint, and then you look at our P haplo deletion predictions, by and large, they do line up really well, but there's going to be a subset of genes, certainly a minority of genes, for which there's some discrepancy between these two models. And that, I think, is getting at the second point that you're raising, which is they're capturing sort of partially independent truth, biological truth, however you want to define that. But in broad strokes, most of these scores are pretty well correlated.
Well, thanks. Yes, we're a little over time, so I think we'll end there. But that was really interesting. Thanks so much for coming.
Yeah, thanks again. Thanks everyone for the attention.
Thanks everyone for joining. See you next time. Have a good day.